Today we will discuss about role of post-translation modifications such as glycans, proteoglycans on the already identified cancer hallmarks. Some of the cancer hallmarks are like growth suppressor, proliferations, and, and this resistance to cell death. All these cancer cells or these tumor promoting inflammations. And all other cancer hallmarks can be found in our previous videos. You can go through the I button and to get more information about these cancer hallmarks. So in this video we will discuss the role of post-translation modifications on cancer hallmarks. We will start with this sustained although these things we have covered separately in many videos, so you can consider it as a summary of what we have covered about the cancer cells hallmarks where this post translation modification plays a role. So now just come back. Now let us come back to our main topic. That is the first panel shows about this sustained proliferative signaling. So in this case, what will happen because of the change in the glycan structures of this decaron and percolon, at the same time this RTK, what it will does is this RTK will trigger the survival pathways such as this P13K AKT. At the same time, this SIMIC, FOXM1 and cyclin D1, which are glycosylated in the cancers, and that will also that will promote this upregulation of these proliferative genes. So this upregulation of proliferative genes is the combined effect of these modifications of this modifications of these factors. Sorry, yeah, modification of these factors in the cytosols. At the same time, the modifications of this modification of this cell membrane proteins such as decorin and also this percolon. Next thing, we will talk about the resistance to cell death. In the resistance to cell death, what will happen? The glycoproteins such as TREL and FAS has these different glycan profiles, and these different glycan profiles, what it will does is it will prevent the activation of this trial and FAS and prevent the down regulations of these signals that can trigger cell death. So those cell death can be prevented or inhibited by the hindered by these glycan structures. At the same time, the aberrant glycosylation on this alpha 3 beta 1 integrins also prevent the cal3 to interact with these integrins, and thus they cannot inhibit these integrins triggered P13 AKT pathways, and this NF kappa beta pathway that also plays a role towards apoptosis is also been glycosylated. So its roles is also reduced towards triggering apoptosis pathways. So overall impact will be that upregulations of only the survival genes or mainly the survival genes, because these P13K AKT pathways are activated and other pathways related to apoptosis are down regulated. Next one is about this deregulated cellular energetics, and this we have covered many times in our many of our previous videos, where glycosylated pathways from glucose is been diverted. It's been diverted in such a way that it does not require mitochondria to generate energy such as ATP, whereas what it will does is it will directly generate energies within this cytosols of the cells, and at the same time it will produce pyruvate that can lead to these lactate formations and lactate secretions outside the cells. At the same time, the hypoxia condition that is formed because of the less amount of oxygen in the cancer cells, it will promote this HIF1 alpha target genes, and that will further aggregates this pathways, this glycosylated pathway. So this is how deregulated cellular energetics or changing reprogramming within the cells helps in maintaining their helps in maintaining the energy required for uncontrolled growth rate. And next one is about this evasion of the growth suppressors. So as we know that one of the characteristics of the cancer cells is the uncontrolled growth rate. So one of the reasons behind this is that the growth suppressors get reprogrammed in a way or glycosylated in a way that they do not do its function. such as the p53, which is an important growth suppressor, such as when the uh, de, uh, when the problems within the cells becomes uncontrollable, then p53 triggered apoptosis of the cells. However, in this case, the p53 functions is dysregulated because of this glycnec attached to this p53, and thereby the cell death will not be cannot be triggered, that can usually be triggered by p53, and that will cause the cell cycle progressions. And in the case of genome instability, what will happen? The genome, all the mutations that are occurs, all the mutated genes that are occurs, there is a self-regulatory mechanism within the cells that it can degrade, or proteasomal degradations occurs of the all the of most of the mutated proteins. However, in this case, because of the mutations of this ATM, those mutations will not be occurred, and that will cause this mutagenic and oxidative microenvironment. Next one is about angiogenesis. So angiogenesis will get upregulated because the VGFR, because of the aberrant glycosylation of these VGFR2 receptors, that is able to interact with this GAL1 and can promote this beta-tinK AKT pathways, and these pathways helps the cell's survival. And whereas this aberrant glycosylation of this neurophin and decorin will promote angiogenesis within this endothelial cells, and thereby helps in providing the efficient food requirement, or the energy requirement that is required by the tumor cells for maintaining the uncontrolled growth rate. Next one is about the invasion and metastasis. In the case of invasion and metastasis, the specific kind of glycan structure, such as this SLE glycan structures, helps in interaction with the selectins, or lectins at the endothelial cells, and thereby helps in the intravasation at the secondary infection sites. And after that, these glycan structures also helps in restating or changing its properties from mesomicals to epithelial cells, and thus now they generate, or now they spread tumors at the grow tumors at the secondary organ sites. And these things we have covered in detail in one of our previous videos. Next one is about this tumor promoting inflammation. So in this case, some of the tumor cells which get exposed to the immune cells will generate immune response, or the inflammations, and the excessive exposure of these non-human epitopes will cause excessive inflammations, and it might even cause some of the side effects. And next or last one is about this immune escape. So because of the aberrant glycosylations, what will happen is the dendritic cells, which has a function to interact with the non-self epitope and generate antibodies with the help of T cells against those epitopes. However, because of the aberrant glycosylation, what will happen is that dendritic cells will not be able to reach to the non-self epitope that are generated on the tumor cells. At the same time, some of the glycan structures also attenuated the NK cells functions, or deactivate the NK cells. So in this case, the immune escape occurs by preventing the dendritic cells and the NK cells to interact with the tumor cells. So this is a general overview about this effect of this post-translational modification on the cancer hallmarks, or how the cancer post-translational modification contributes to this cancer hallmarks, not hallmark, cancer hallmarks. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.